Hi guys, this is MedTutor Barry and welcome to my channel. In today's video, we're going to be discussing G-protein coupled receptors. This is a follow-up video to my video on the overview of cell membrane signaling. So in this video, we're going to be discussing the big G-protein receptors, the major types, which include Rgq, Rgi, and Rgs proteins. And we'll also be discussing small G proteins briefly, and then we'll end with discussing a specific second messenger system that is quite high yield. It's called cyclic GMP, and we'll end with discussing about cyclic GMP. So let's get started. So let's start with Rgq G-protein. Okay, if you guys remember, cell membrane signaling, there has to be a ligand or a messenger that comes from the outside world. It has to be received by the receptor, and then there has to be downstream signaling within the cell to cause some kind of a cellular change. So same thing here. When a ligand comes and binds to my receptor that's associated with the Rgq G-protein, that Rgq G-protein gets activated. Okay. So the G-protein has three subunits. It has an alpha, a beta, and a gamma subunit. When the ligand comes and binds, this alpha subunit gets phosphorylated. So GTP drops off a phosphate to my alpha subunit, which turns on my alpha subunit. Okay. Upon activation of my G-protein, these subunits dissociate. Let's draw that out. My alpha goes here. Beta and gamma go together. The alpha subunit, which is turned on, goes down into the cell. Now this alpha subunit associates with an enzyme called phospholipase C. Okay, phospholipase C is a phospholipase, which is an enzyme that cleaves phospholipids. Okay, so lipase, it cleaves phospholipids. So just kind of a side note here, remember that our phospholipids are nothing but a glycerol head attached to two fatty acid chains. There's a phosphate on my glycerol, which is connected to a head group, in this case inositol, because what we're talking about is PIP2. Okay, this structure here is PIP2. Phospholipase C goes in and cleaves this phospholipase at this position between the glycerol and the phosphate head. So what I'm left with this glycerol and the two fatty acid chains, which we call our diacyl glycerol. And I'm also left with the inositol head group with the phosphate, which we call IP3. Okay, so once phospholipase C is activated by my alpha subunit, it helps to cleave BIP2 into IP3 and DAG, which we're also going to represent by the glycerol and the two heads. This DAG is actually sent back up to our plasma membrane. Okay, so this DAG travels all the way back to the membrane. This IP3 goes down into my cell to my sarcoplasmic reticulum and opens the calcium gated channels, or the calcium channels. Once IP3 opens the calcium channels in my sarcoplasmic reticulum, the calcium flows out and gets accumulated in the cytoplasm. Now this calcium acts as a second messenger, and it can do a bunch of different things. Okay, we're going to discuss a few in this video. One of the first things it does, and one of the more important things, is it can associate itself with, actin and myosin in muscles, and it can cause muscle contraction. Okay, so it causes muscle contraction by associating with the actin-myosin complexes. Another thing this calcium can do is it can associate with calmodulin, and form this calcium calmodulin complex, which can go down to do a bunch of things like turn on certain kinases. Sorry about that. So in this video, we're going to see how kinases, we're going to talk about some specific kinases when we get there. And also we're going to talk about how they go to our DNA, how they can turn on transcription factors, which can go to our DNA and affect gene expression. Another thing calmodulin can do is it can turn on this enzyme called nitric oxide synthase. We're going to talk about nitric oxide synthase at the end of this video when we talk about our cyclic GMP as a second messenger. But I think this should be enough for now. And a third thing that calcium can do is it can go up, sorry about that, it can go up to my DAG, and calcium and my DAG together can turn on a specific protein kinase called protein kinase C. Okay, this is our first specific protein kinase that we're looking at. And like I mentioned earlier, protein kinases have the ability to turn on transcription factors. In this case, one of the big ones it turns on is NF-kappa B. NF-kappa B goes to my DNA and eventually impacts gene expression of certain genes that affect our cells, control our cell cycle, etc. Okay, so that's everything not everything, but most, a lot of the things that are GQ associated receptor can do within the cell. Okay. We're going to move on to our GS and GI. It's really easy to understand these because they're the exact opposite of each other. Okay, let's do GS first. GS stands for G stimulatory. GI stands for inhibitory. The I is for inhibition. And we'll see that when we get there. So GS again has an alpha, beta, and gamma subunit. Once the ligand binds, the alpha subunit gets turned on or activated by GTP donating a phosphate. This is then going to the subunit split. The alpha subunit, which is active, goes down and turns on a specific enzyme. And in this case, this enzyme is called adenylate cyclase. Okay, adenylate cyclase. Now, adenylate cyclase is an enzyme that helps us take ATP and convert it into cyclic AMP. Now, cyclic AMP is a very, 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 very important second messenger. Okay, and you're going to see this everywhere. So adenylate cyclase is an enzyme that can take ATP and convert it into cyclic AMP, which is a really important second messenger in the system. Cyclic AMP can actually go down and turn on a specific kinase called protein kinase A. So this is our second specific kinase that we're talking about in this video. Okay. Protein kinase A, like most kinases, can turn on specific transcription factors. In this one, a big one that it loves to turn on is CREB, and CREB can go to the DNA and alter gene expression. Okay. GI is just the exact opposite of GS because GI likes to inhibit things, right? So our alpha, beta, gamma, alpha is turned on, GTP comes and drops off this phosphate. The alpha subunit of my GI inhibits adenylate cyclase, which inhibits cyclic AMP, which inhibits protein kinase A, and which inhibits transcription factors from eventually inhibiting gene expression. Okay, so GI plays an inhibitory function in my downstream signaling. Okay, we're going to move on to talk about some small G proteins very quickly, and then we're going to move on to discuss our second messenger system called cyclic GMP. So small G proteins are G proteins that have their own GTP and GDP. So they have an intrinsic GTPase activity. They constantly switch between a GDP associated off state and a GTP associated on stage. Okay, they're constantly kind of switching back and forth. They have intrinsic GTPase properties. These are they play an important role in our cells. They get turned on by certain signals, and they keep kind of flip-flopping between these. So one important example of a small G protein is our Ras protein, if you've heard of that. Ras plays a key role in controlling growth factors in our cells. So Ras is also considered an oncogene because if Ras stays on indefinitely, that can give you cancer if you're not able to turn it off and regulate growth of the cell, right? So this is one of the oncogenes, one of the examples that you can that is discussed in medical school. So how does this small G protein go from an off stage to an on stage? It can do that by acquiring a phosphate. Now, in order to acquire phosphate, it needs a catalyst, and the catalyst to help it turn on is called GEF. Okay, this is the enzyme that helps put that phosphate onto my GDP and convert it to a GTP. We have a catalyst to do the opposite, which is called GAP, and this removes a phosphate, converting my on stage back to an off stage. Okay, so these are small G proteins with intrinsic GTPase activity, and one of the examples is Ras. Okay, so this is a summary of my GQ, GS, GI, and small G proteins in my cell. Let's just quickly talk about another second messenger called cyclic GMP. So if you guys remember, we just talked about this messenger, really important messenger that I stressed on, called cyclic AMP, right? Adenylate cyclase turns on, is able to convert ATP to cyclic AMP. Cyclic AMP turns on protein kinase A, right? Similarly, we have a second messenger system. So let's try to draw this line just to show you that this is happening in another cell, and I'll show you why that's important in a second. We have this other enzyme called guanylyl cyclase. Just like adenylate cyclase, we have guanylyl cyclase. This, once it's turned on, is able to take GTP and convert it to cyclic GMP. Okay, just like adenylate cyclase took ATP to make cyclic AMP, this is taking GTP, let me just rewrite this, to make cyclic GMP. Cyclic GMP goes down to turn on protein kinase G, which is the third specific kinase that we're talking about today. Okay, protein kinase G. Now, protein kinase G has a bunch of different functions. One of the big ones that we're going to talk about is, sorry about that. One of the big ones we're going to talk about is muscle relaxation, especially in the blood vessels. It helps to relax that tunica media layer in the blood vessels. It helps the blood vessels dilate. It also helps with platelet inhibition, and it can also do or control some gene expression. Okay. But let's go back to the example of muscle relaxation, okay? And specifically in my blood vessels, causing my blood vessels to dilate, Okay, so in order for my blood vessels to dilate, I need cyclic GMP. Hopefully that makes sense from this pathway. Now, who is able to turn on the starting point, guanylyl cyclase, right? Who does this? So to understand that, we have to go back to this nitric oxide synthase that we talked about when we talked about our GQ proteins. Okay, so let's look at what a blood vessel looks like. A blood vessel, keep in mind that there will be layers of muscle, but on the very inside, or the most luminal side, I will have a layer, a single, if you can see the yellow, this is my endothelium. Okay, this is my endothelium, is the yellow, and the muscle there is around the endothelium. My endothelial cells are the ones that are going to make this nitric oxide synthase. Okay, so this nitric oxide synthase, this whole GQ pathway, GQ turning on phospholipase C, turning on IP3, giving us calcium, turning on calmodulin, giving me nitric oxide synthase. This whole pathway would happen in, or would take place in the endothelial cells of the blood vessel, which is this layer here on the luminal side. This nitric oxide synthase would actually go down and do some more downstream signaling. And specifically what it would do is it would take our amino acid arginine, and it would help us convert arginine to citrulline, and in the process release nitric oxide. Okay, let me just rewrite that so it's more clear. Nitric oxide synthase would help convert arginine to citrulline, and in the process release nitric oxide. Remember that nitric oxide is being made by the endothelial cells. Okay, just reiterating that. This nitric oxide can actually go then enter from the endothelial cell into my muscle cell, and this here, let's make a note here. This is my muscle cell, or my smooth muscle cell in my vessel. This nitric oxide goes from my endothelial cells into the smooth muscle cells, and this helps to activate guanylyl cyclase in my smooth muscle cells, right? Once guanylyl cyclase is activated, cyclic GMP is activated, which can turn on protein kinase G and eventually cause vessel dilation. Or dilation of the muscle. Okay, so that's the cyclic GMP pathway. It's a really important second messenger system that you're going to see in a bunch of places. And don't confuse this with cyclic AMP, right? And another thing that I would like to quickly mention in the end is both cyclic AMP and cyclic GMP are degraded. Okay, they're both degraded. Not sure how to write this, but they're both degraded by these things called phosphodiesterase. Okay, phosphodiesterase help with degradation of cyclic AMP and cyclic GMP. Okay, there are certain drugs that we can give, and they're called phosphodiesterase inhibitors. This is Q for pharmacology. Phosphodiesterase inhibitors inhibit phosphodiesterases, as the name suggests, and help us keep the levels of cyclic AMP and cyclic GMP up in our cells. One example of this is sildenafil or Viagra. So in the blood vessels of the penis, we want to try and keep cyclic GMP levels up to cause more vasodilation for longer, so more blood can fill up. So one of the mechanism of action of the drugs is to block those phosphodiesterases, specifically PDE5, which can keep the cyclic GMP levels up and give us prolonged vasodilation in the blood vessels. Okay, so that's a pharmacology cue for you here. So this is the summary for overview of GPCRs. I hope this helps you put everything together and makes a little bit more sense. And thank you for your time. I'll see you in the next video.